Good morning everyone. I am Brindha. Today I am going to discuss about marine microbial community protozoa. Marine microbial community is the vast community which contains very large microbial diversity and flora and fauna. As the beginning, we want to see what is protozoan. Here is a small quiz that who am I? I am single cell but not a prokaryote. That means it is a eukaryote. Have false leg, but they call me as an animal. It is known as animal-like structure. It have false leg, that is pseudopodia. It containing the membrane-bound nucleus, so that it is named as a eukaryote and animal-like structure. It can be it cannot be prepare their own food and act as a parasite, and it always feed on bacteria. This is the common things that we want to know about protozoan. Protozoan is the primary consumer which always consume bacteria, and zooplankton that consume this protozoan in the marine environment. And these protozoan was a sediment deep in the oceans that was used for the many research studies that we can see in the next slides. Here you can see some of the microscopic structure of the protozoan, and this side you can find the small star-like structure. This star-like structure is nothing but the sediments of protozoan, which is taken from Japan, and they found that it is 500 million years old protozoan sedimental shells. So that these protozoan are mainly used in the research and also the finding of the previous finding or dating of the environment and climatology in the history. Yes. And this is common classification of the protozoan, and the protozoan was from the kingdom Protista, and it can be amoeba, flagellates, sporozoa and ciliates. Here, more commonly, when we studied about protozoan, ciliates come and play a major role. Ciliates is nothing but, as it represents in the name, the whole body is surrounded by a small hair-like structure known as ciliates, and there are more than 7,000 species in existed in the world. And the ciliates containing the two types of nucleus. One is macronucleus and this micronucleus. Macronucleus is stored for the nutrient and food, and smaller nucleus, micronucleus, it's used for the reproduction. And the major one is contractile vacuole. This contractile vacuoles is present only in the protozoan which live in pond, river and freshwater environment. The protozoan which is live in the marine environment lack this contractile vacuole, because in marine environment it is the hypertonic solution which is the very high salt content. When this contractile vacuole present, this hypertonic environment make the water present inside the cell that expelled through this contractile vacuole, and it will affect the cellular growth and its metabolism, so that the contractile vacuoles was absent in all the marine protozoan animals to save themselves from this hypertonic environment. In this protozoal diversity, we are going to discuss about few major families that was present in this marine environment. The first one was foraminifera. This is commonly known as foramen, which is nothing but it containing a specialized shell covering known as testa, which containing the chambers. That chambers were connected through the valves. This testa is made up of the calcium carbonate, and it also containing the cytoplasmic flow from both bidirectional from inside to outside and outside to inside. Then these are all the different structure, which is in the first picture we can see here, it is the different structural arrangement based on the tests, that is that is the shell made up of. And the second one is known as the different arrangement based on the aperture. Aperture is nothing but it is like a mouth-like opening which is surrounded by small cilia's or tentacles that will help the protozoan to engulf the food. Then here it is the reproduction. It can carry out both sexual and asexual reproduction. Here we can see that the haploid young spores which is formed into schizoid form, which is leads to the asexual reproduction. And this after asexual reproduction, it will undergo the mitosis and form gamete. These gametes were fused together to form a zygote, and these zygotes were then undergo into a diploid microsporic that will again undergoes into meiosis, and that produces the haploid young gametes, so that this foraminifera can also perform the sexual and asexual reproduction. Contributions in marine ecosystem. As I said below, the foraminifers form form as sediments, and it acts as the primary producers, grazers, and also the decomposers. First thing when we saw in the few slides before, that is a grains of stores that was isolated in Japan, is this foraminifera cells, and they used to find they used to find that temperature and environment that how existed before few million years before. That technique is known as paleoceanography or paleoclimatology. They use the shells. How they use the shells? Normally, they use a kind of ratio known as oxygen isotope ratio. The oxygen can exist in two forms. First one is O16 and O18. O16 is a light weighted form, and it can easily precipitate and evaporate. O18 is comparatively stable than O16. During the winter climate and glacier formation, this O16 present in the seawater will automatically form a sediment or precipitation in the ocean, and in that time O18 is available in a large amount in seawater. And in the summertime or in high temperature, the glaciers that formed was melted and released this O16 into the salt water. Then the O16 content will overtake this and increase then the O18. That is the oxygen isotope ratio. How they will detect this oxygen isotope using the shells of foraminifera, that is these organisms either present in cold environment or in the summer or hot environment, by determining the utilization of O18 or O16. If they are present in the cold environment, they will utilize O18. If they present in the hotter or warmer environment, they will utilize this O16. Using this, the play the paleoclimatologist determine the climate in the climates during the climates in few decades before, and it is also act as an indicator species depend upon its in abundance and distribution. And it can also use it to determine the water quality, and it can use it to see how the environmental factors present in the ocean. And it also used for oil and gas exploration based on this oxygen and oxidization. The oxidization of these This sediments leads to the formation of fossil fuels, and these fossil fuels can lead to the formation of oil and petroleum. Using the several advanced technologies, we will trace the foraminiferous abundance in the marine environment, and we will use that thing for the gas and oil explorations. The next family is Radiolaria. It is nothing but silicaceous microprocesses. It is also a kind of holoplanktonic protozoan, which is nothing but the center region. It is hollow. That is a central tube which is hollow, and it containing it is non-motile, but it has the shell that will lead to the buoyancy movement of this radiolaria, and its skeleton mainly made up of strontium sulfate, which is easily dissolved in water. But this was replaced in future by silicon. When it replaced by silica, it is changed into now changed into sediment that can have high geographical range, and it can stay for a long period, and it can present even carbonated compensation depth, and it is mainly useful for the research studies and paleoceanography and paleoclimatology. And here we can see the morphology, that is the radiolaria containing the two parts, that is capsule and polypide, and the polypide is the mouth of opening which containing the tentacles or small cilia-like structures, and it undergoes binary reproduction, and it can either live in a colony or separate individual species. It can occur in a different colors. Next, the important phenomena by radiolaria was biomineralization. Biomineralization is nothing but the living organism that absorb the mineral and store that minerals in the organic matrix, that will lead to the formation of the inorganic shell cells. These radiolaria will absorb the silicoic acid, that is orthosilicoic acid, that is in a dissolved form in seawater. It absorbs those acid and stored in its vacuole. Once it reaches the threshold, it will next transfer the silicoic acid into silica, and using the special transporters, this silica was exported out to the ectoplasm. As we know, ectoplasm is the outer layer. It transported to the ectoplasm. There it made a silica shell. This will act as the protector, and it will prevent this radiolaria shell from the deterioration, or this silica cell will prevent from easily dissolving in the seawater. But sometimes due to the process of making or uniqueness of the different different uptaction of this silicoic acid, sometimes it will degrade and dissolve, and that degradation and dissolving of silica will play the major role in the silica biogeochemical cycle. Radiolaria bioluminescence. This phenomenon is mainly used for the communication, defense, and also sometimes for reproduction. As we all know, the bioluminescence, the major enzyme is luciferase, and it also used other enzyme called as photoprotein. This enzyme will bind to the cofactors, and it will emit the energy that will lead to the formation of the bioluminescence. And environmental indicators, as I discussed about the orthosilicoic acid formation and the silica cells, that is known as opaline silica skeletons. These opaline silica skeletons was mainly found in the four species, as I mentioned here: coral, Colodaria and Spumellaria and Orodaria and Nassalariasis. This was highly resistant to the dissolution in the seawater, and it mainly used for the environmental reconstruction and paleoclimatology. And photosymbiosis. Photosymbioses is as normal symbiosis, whether this protozoan radiolaria is having a symbiotic relationship with algae, where the algae uses the photosynthesis and provide food to this radiolaria, and in back it will provide the nutrient rich environment for the algal growth. Ciliates, as in the name, these things containing the ciliaceous that used to flow in the water current for locomotion, and it feed the bacteria, and its main role was feeding bacteria and organic matter and helps in the nutrient cycle. It's the major role of this cilia, and it is common. It is a major contribution of the ciliates was the secondary invaders of the coral reefs. These cilia can be three types in the marine environment: either free-living or crawling ciliates or stalked ciliates. Free-living that possess the cilia in all over the surface of the body, and crawling ciliates only present in the ventral surface and the belly region, and stalked ciliates is nothing but the cilia present only near the mouth and present in the head. Here you can see the different stages of ciliates that was isolated from the coral reefs. That the first one was the adult stage, that is ADEG, and the second one was the larval stage BEHK, and the third one is the divisible or during the reproductive phase, that is binary fission phases CF and I and K. These are all the different stages of cilia that was isolated from the coral reefs. As I said before, these ciliates play a major role in nutrient cycle, and it also known as the secondary invaders of the coral reefs. In the heterotrophic nutrients, it will excrete the carbon, nitrogen, phosphorus that take part in the nutrient cycle, and in the mixotrophic nutrients, it will release carbon, nitrogen, phosphorus, and dimethyl sulfur propanate. These the mixotrophic nutrients is nothing but both autotroph and also heterotroph. These nutrients excreted by these ciliates will take part in this nutrient cycle, and in the coral relief, that is these coral reefs will excrete the mucus. This mucus will attract the symbiotic microorganism that will prevent the entry of the pathogenic microorganism into the coral reefs that will accumulate inside the cells. These are all known as symbiodinases. These symbiodinases was present inside the cells. During the high temperature, the nutrients present in the coral reefs also decreases. So this symbiodinase was expelled from these coral reefs. After its expulsion, it present in the sea water. During that time, the nutrients released from the ciliates, that is especially this DMSP, which is mainly served as the sulfur source for this symbiodinase, and the carbon sources released by the ciliates were taken by the symbiodinase. After taken all these sources that released from the ciliates, then again it was returned back into this coral reef, and that is coral resilience. After it returned back, it will lead to the formation of an antimicrobial compound, that is TDA, that prevents the entry of the pathogenic organisms into the coral reefs. So that the ciliates plays the major role in the coral resilience and recovery of the coral reefs from the unhealthy environments and prevent it from the pathogenic entry of several pathogens. Then ciliates is the bioindicator. It can used in the artificial wastewater treatment plants as an indicator while treating these wastewaters, and also it is frequently the environment was frequently monitored by testing these ciliates, whether it can affected or accumulation of toxic matter, heavy metals and detergents. And the next one is bacteriovirus. Bacteriovirus is nothing but normally we know these ciliates will feed on bacteria. So during aquaculture, they will use these ciliates and several protozoans to feed on the bacterial species that presents in the aquaculture during the culturing of fishes. Apicomplexances is nothing but the intracellular parasites. It also comprises of the parasitic organism that we well know, that is malarial parasite, plasmodium, and etc. These apicomplexances also affect the marine species like marine invertebrates. When this apicomplexan that infect marine invertebrates known as marine gargarians, and when this apicomplexan affect the coral reefs, that is known as corallocoids. This is the current paper that is published in 2024, that they found that the coral infection by this apicomplexans was very large in the cold marine ecosystems. So these are all the protozoan families that was present in the marine environment, and these four families are dominantly present in this marine community. Due to the time limitation, I can deliver the few information, but depend upon the protozoa, many researchers and many dating of years and many studies were still going on based on the sediments and their fossil fuel of these protozoans, and several symbiosis association with many other marine species. So that as we concluded, we need to protect and restore ecosystem, especially marine ecosystem, because each and every and single microorganism in this marine ecosystem plays a major role, and it can create a great history. When we were in the future, when we turn back and see, these are all the path that we leave behind for our generations. We can use these things, protozoa, many things for calculating how we are living, how we impact this society and microbial community. So that these are all the things about protozoan in this microbial, marine microbial community. Thank you.